So the second hour of today's lecture, we're going to talk about the dynamics of genes in the population. If you have taken population genetics, this chapter should be relatively easy for you. But if you don't, I think for most of the part of this chapter, you have learned it from, say, general biology, because we are going to talk about Hardy-Weinberg equilibrium for the beginning part of this lecture. So just try to define, and I also think you already know that, a species is defined as a group of organisms potentially capable for inbreeding, and that was defined based on the biological species concept. And a population is a group of organisms of the same species. So the individual of the same species in a particular region, we call it a population. So when we concern about multiple species, we don't call it as a population, even they stay in the same location. And DIM is a particular isolate interbreed, like a small population. So this term will be used when we concern about the population structure. If population will have small isolate groups, then we use this terminology. And in evolution, we particularly interest in a life cycle of the organism. So when we say a life cycle, it's from the adult produce gametes, then the gametes will form the zygotes for the next generation. So usually we're concerning about the generation to generation, particularly the changes between generations. Remember last time I asked you what's the definition of evolution, and one of your classmates say it's the changes like through time. And in population genetics, we usually concerning about like from one generation to another, but also the later part of the generations. But we always start from one generation to the next one. And we like to ask, what is the equilibrium? So all the terms will be used in the population genetics. Okay, so this cartoon just to show you in the view of a population geneticist, actually it's not only a group of organisms, but a group of organisms which carries the genes, the alleles in their genome. So we are concerning about a population, and of course all the alleles are assembled within the individuals. So showing here just to show you is if it's one locus with two alleles, A1 and A2. And all the individuals here are either two different homozygous or the heterozygous A1, A2. And that's the overall gene pool we are concerning about. So this is the view of the population. There are individuals, but for each individual, we are more concerned about their genotype of this particular individual. So that is showing in this one. And the calculation is relatively easy because we just want to know the frequency of each allele. That's the A1 and A2. And also the frequency of each genotype. That's the A1, A1, A2, A2, and A1, A2 showing here. So this is just like the numbers you can count from the population. This is a very simplified version. So you can count from the population and calculate the frequency. So usually I will ask you the allele frequency. So the allele frequency is A1 or A2, and or the genotype frequency. That's the A1, 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 A2, and A2, A2. And just keep in mind, and I think you also know, if we say the frequency A1 equal to P, and the frequency A2 equal to Q, and the population only have two alleles, so P plus Q equal to 1. So that's no exception. So So that's something you have to keep in mind, because later when we do calculation, the probability, the sum of all the probabilities equals to 1 is like the general rule. So this is the simple model. And how to calculate the frequency. This is like all the words, but you already know it. If you know how to calculate the allele frequency from the previous slide, you already know the rules. So the rule is the number of one particular allele divided to the total number of alleles, right? So if take the A1 as an example, you want to count how many individuals carry A1 and how many A1 alleles for each individual, right? So if it's A1 homozygous, that particular individual carries 2 A1 alleles. So that is like a very simple calculation. Then total number of alleles is also a simple. How many individuals in the population we surveyed? If we survey 20 individuals, each individual carry 2 alleles for a common autosomal inheritance like a gene. Then we have 2 times 20, so that's 40 in total. So that's the total number of alleles. Then you also can calculate the genotype frequency. Then that's also very simple. You just count how many, for example, here I use the MM locus. So how many individuals is MM homozygous, how many individuals are NN homozygous, and how many individuals are MN heterozygous. So those are the numbers, and you divide the total, for example, 200 in this example, then you can calculate the genotype frequency. And the allele frequency also can be calculated from the genotype frequency as shown in the next slides here. So because we already know the homozygous carry 2 alleles, the same alleles, like MM has like 2 M allele, NN has 2 N allele. So the frequency of M is contributed by both MM homozygous and MN heterozygous. So this is like showing on this slide, and just like also a simple calculation, you can say, so the frequency of M equals the frequency of MM genotype, and the half times the MN genotype frequency. So that's like how many alleles contribute by this particular genotype. So that's the alternative way to calculate allele frequency. The same thing you can calculate frequency of N, so the rule is the same. So that's the simple calculation for allele frequency. I want to remind you about alleles. I found this is rather difficult because every time if I ask what is allele in the exam for genetics, for population genetics, occasionally in evolution or in general biology, and most of the case I have half or even more than half of students get the answer wrong. So just to remind you, the definition of allele is the alternative form of a gene. So when we say alternative form, if we take a simple example as our ABO blood type, so you know we have A type, B type, and O type, that's the null allele. So we have A allele, B allele, and null allele. That's the 3 common alleles in human population. And the combination of the 3 will have different genotypes. But usually when we talk about type, we're talking about phenotype, right? I'm an A-type individual, but I don't know whether I'm a homozygous A or a heterozygous A, whether I have a null allele in my genome, which I don't know. But the 2 alleles are different at the sequence level. We call that different alleles. And I think if I use this example to ask you, given the human population have four different phenotype, I forgot AB type. So A, B, O, and AB type. Then there are 3, and I ask you how many alleles, or I say we have 3 different alternative forms of this gene. That's A, B, and the now allele. Or I didn't say allele, I say A, B, and the now. Then I ask you what's the terminology of this. You probably always say alleles, especially if it's a multiple choice question. But when I ask you the alternative form of a gene called what? And a lot of people doesn't really know what I'm asking for. And some people hate to have this like a definition question. But it's not I like the definition question type of question, but I just found the allele become more complicated when we learn the sequence. Why do I say so? Because at the sequence level, we see more nucleotide difference, more changes. So actually, if we look at the sequence, the same ABO type gene, then you will see there's more alleles. So sometimes in your textbook or in a paper will say this is A-classed alleles. Means all the changes in the sequence still give the final phenotype as an A type. Then that's we call it A-class alleles. But that only counts when we look at the sequence, right? So any changes at the nucleotide, at the DNA sequence level, and we call that different alleles. So I think that's the difficult part for most of us. When we talk about alleles, then what do we mean by alleles? Any differences at the sequence level, we can call that different alleles. So that's what I want to remind you.